Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about Krebs cycle. So Krebs cycle, it is in the aerobic respiration. After link reaction it will start. It is also called as citric acid cycle because of the first product it produces, and Krebs cycle because based upon the scientist name, or TCA cycle, that is tricarboxylic acid cycle. So it is a series of reactions that takes place in the mitochondria, results in the oxidation of acetyl CoA to release carbon dioxide and hydrogen atoms that later lead to the formation of water. Okay, so because of the first product we are calling it as a citric acid cycle, as the first metabolic intermediate formed in the cycle. Okay, that is citric acid. This cycle is termed as tricarboxylic acid because of the presence of the carboxylic acid molecules. Next to the citric acid cycle is the first final common pathway for the oxidation of all the biomolecules, that is fats, proteins, carbohydrates, everything. So that this will enter into the cycle through the acetyl CoA. The citric acid cycle is a cyclic sequence of reactions with the 8 mediator reaction. The cycle is also participating importantly in it provides the electrons or high energy molecules to electron transport chain for the production of ATP in the water. Pyruvate, which is formed at the end of the glycolysis, is first oxidized into acetyl CoA, which is a 2 carbon, which then enters the citric acid cycle. So 2 pyruvic acid will produce 2 acetyl CoA. It will enter the cycle. So where it takes place? So in the eukaryotes it takes place in the mitochondria, while in the prokaryotes it takes place in the cytoplasm. Okay, the pyruvate formed in the cytoplasm is brought into the mitochondria where further reactions will take place. The different enzymes which are involved in citric acid are located either in the inner membrane or in the matrix space of the mitochondria. So in the prokaryotes mitochondria is absent. So that's why these reactions are taking place in the cytoplasm. So let us see the reactions which are involved in the Krebs cycle. First one is the condensation. So the first step of the citric acid cycle is the joining of 4 carbon compounds oxaloacetic acid with the 2 carbon acetyl CoA. So as oxaloacetic acid react with the acetyl group of the acetyl CoA and water results in the formation of 6 carbon compound, that is the citric acid. So this reaction is catalyzed by a special enzyme, that is the citrate synthetase. Citrate synthetase catalyze this reaction to produce the citric acid. So that's why we are calling it as a citric acid cycle. So oxaloacetic acid, it is a 4 carbon one, and acetyl CoA, it is a 2 carbon. So that's why 6 carbon molecule is formed. If you see here, acetyl CoA is mixing up with the joining with oxaloacetate or acetic acid and giving rise to the citric acid in the presence of citrate synthetase. In the presence of citrate synthetase, this is taking place. Next reaction, dehydration. So that is the removal of the water. So citric acid is converting into the cis-aconitase. So this reaction is catalyzed by the enzyme aconitase enzyme. This reaction takes place by dehydration of citrate to the cis-aconitase. And the next reaction, that is the hydration. So hydration means addition of the water. Here it is removed, and in the next reaction water will be added. So cis-aconitic acid will form into isocitric acid. Citric is converting into 6-aconitase. This reaction, actually cis-aconitic acid converts into isocitric acid in the presence of water, and the same enzyme aconitase will participate in this reaction. Okay, this is about the hydration. Now isocitric acid will undergo the reaction. So that is the oxidation reaction. So the next reaction is the oxidation reaction. So in this reaction, actually oxidation is taking place in the aerobic respiration. In the presence of oxygen, the reactions will take place, right? So here isocitrate, that is the 2 molecules of the isocitrate on the oxidation gives 2 molecules of oxalosuccinic acid. 2 hydrogen removed in this and accepted by the 2 NAD plus in presence of the isocitric dehydrogenase enzyme, and it will produce 2 NADH molecules in this reaction. So because of this, isocitrate will converting into the oxalosuccinic acid and 2 NADH are formed. So if you see the reaction here, you will find so 2 NADH molecules it will form. So the next reaction after the oxidation, the next reaction, let us see, oxidative decarboxylation. Oxalosuccinic acid, which is a 6 carbon, is oxidatively decarboxylated. That means removal of the carbon is taking place from this 6 carbon molecule to form 5 carbon compound, that is alpha ketoglutarate acid, which is catalyzed by the enzyme oxalosuccinic decarboxylase, and it will release 2 carbon dioxide because 2 oxalosuccinic acid, so that's why 2 carbon dioxide are formed. Okay, so alpha ketoglutarate acid is formed in this reaction. Next, oxidative decarboxylation of alpha ketoglutarate. This is another step of the reduction reactions where alpha ketoglutarate, which is a 5 carbon one, is oxidatively decarboxylated to form a 4 carbon compound, that is a succinyl CoA and carbon dioxide, because here 5 carbon one, again the carbon is removing and becoming the 4 carbon molecules, so carbon dioxide is removed. The reaction irreversible and catalyzed by the enzyme alpha ketoglutarate dehydrogenase. It is found in the mitochondrial space. This reaction is similar to the oxidative decarboxylation of pyruvate involving the reduction of NAD plus into NADH. So here CoA addition is taking place, carbon dioxide removal and also product formation. Next, substrate level phosphorylation. In the next step, succinyl CoA undergoes an enzyme energy conserving reaction, which succinyl CoA is cleaved to form succinate or succinic acid. This reaction is catalyzed or accompanied by phosphorylation of GTP. That is GDP is converting into the GTP. Guanosine diphosphate is converting into the guanosine triphosphate, which is equal to the ATP. Later it will convert into ATP. This reaction is catalyzed by the enzyme succinyl CoA synthetase. Succinyl CoA synthetase will participate here. So succinyl CoA is converting into the succinic acid, which both are the 4 carbon ones. Next, oxidation. The 2 molecules of succinic acid on oxidation gives 2 molecules of fumaric acid. So 2 hydrogen removed in this and accepted by the 2 FAD plus, so they will become 2 FADH molecules, and this is taking place in the presence of succinic dehydrogenase enzyme. So here fumaric acid is formed after this reaction, which is also a 4 carbon molecule. Next, hydration. The fumaric acid or fumarate is reversibly hydrated to form the malate in the presence of fumarase enzyme. As it is a reversible reaction, the formation of malate involves hydration. Okay, so here malic acid is formed. In the next one is the last step, that is the dehydrogenation. So last step of the citric acid cycle. Here oxidation reduction reactions will take place where malic acid or malate is dehydrogenated to oxaloacetic acid in the presence of a malate dehydrogenase, which is present in the mitochondrial matrix. This is a reversible reaction actually involving oxidation of malate and reduction of the to NAD plus into N2NADH. Oxalosuccinate thus formed allows the repetition of the cycle, and NADH formed participates in the oxidative phosphorylation. This reaction completes the cycle. So this is the end, and this will continues. Again this oxaloacetic acid will bind with the acetyl CoA and the cycle will continue, Krebs cycle, and it will produce the carbon dioxides, water molecules, etc. Fine, if you see here the reactions, so oxaloacetic acid is converting into the citric acid in the presence of acetyl CoA. So this will continues at each turn of the cycle, that is per 1 acetyl CoA, 3 NADH, 1 FADH, 1 GTP and 2 carbon dioxide will be formed. But here 2 acetyl CoA molecules are there, so they will be doubled. Thank you for watching.